Okay, hi everyone. So let's move on to our discussion about how certain types of pathogens like viruses and bacteria and parasites have come up with very clever ways of counteracting our immune system, oftentimes coming up with really sort of diabolical sort of means of evading detection, uh, often at the expense of triggering very strange types of immune reactions. So first off, I wanted to take a moment. I meant to do this after our last exam. So I meant to do this on Monday because, after each exam, I'd like to kind of start off the next set of lectures with kind of a look ahead, right? So that you guys know what's in store, so that you sort of have an impression about where we're headed in terms of the things that we're going to learn about in the course. So I'll just do it today since I didn't do it on Monday. So here we are in week 12. So we're at lecture 27. We'll spend a few lectures, 3 lectures, talking about immunity and disease, these examples of how certain types of pathogens create real challenges for our immune system. And I think that's a good way for you, as you have learned all this stuff that we've been talking about our innate and adaptive immune system, to really see some sort of real world examples of these dynamics of how pathogens and our immune system are constantly sort of in this battle of trying to co-evolve. We have, hard to believe, 4.5 weeks of the semester left. So this takes us into the last third of the semester, where I think it's the more sort of fun part of the semester, right? You spend a lot of effort, a lot of energy learning about all the foundational stuff that goes into the innate system, the adaptive system, and we really save a lot of this applied stuff to the end. I think that in past semesters, I found that students really like this stuff at the end because we talk about things like allergies and the role of our immune system in allergies. We talk about things like the challenges faced with transplanting tissues, whether it's blood transfusions or solid tissue transfusions, and how our immune system, ironically, because it's so good at being an immune system, ends up getting in the way of having successful operations between donors and patients. We also talk about things like immunodeficiencies and autoimmunity, right? Very interesting topics. And we wrap up the semester talking about the role that our immune system plays in keeping cancer at bay. Why is it that we don't all just develop cancer as part of our lifetime, right? And it has a lot to do with our immune system. Right? So our last class meeting will be the first week of May, May 5th. That will be a Wednesday, and then we'll review for our third exam. We don't have a real final exam in the traditional sense, right? What we do have is our third midterm exam. Right? So that's how we've been breaking our semester up into basically thirds. So the third midterm exam will be given at the date and time that the final exam would be given for our class. All right. So in other words, I want you to make a note of this, that our final class meeting is Wednesday the 5th, right? And we'll review for the exam. And then the following Monday at noon, that is where, the final exam schedule assigns our particular class as being the time for the final. But instead of having a cumulative final, we'll just have our third exam at that time. All right. And it'll be the same format as the previous two. It's not going to be any longer, right? But it just means that it'll take place at that date and time, Monday the 10th at noon. So please keep that in mind. Those of you who may have already been sort of making plans for after when school is done, if you're going to travel or something like that, be aware of that. Okay. All right. Let's go ahead and start in with our discussion about immunity and disease. So I think it comes as no surprise to you, based upon all the discussions we've had so far in the class about how different pathogens are always mutating and evolving and changing things like antigens that they present, how that can provide a constant challenge for our immune systems to be able to keep up. Thank goodness we have such a good adaptive immune system in order to be able to deal with that, right? Otherwise, in the course of our lives, the first time something comes along that our innate system doesn't come pre-programmed to deal with, that would be the end of us, right? But because we evolved to live many decades of life, 80, 90 years or even more, we have evolved a strong adaptive immune system to be able to get us through those many decades, right? Being able to, yes, get sick from pathogens that surprise us, but ultimately we expect our adaptive immune system to be able to come up with a solution, right? And that relies upon things like selection of B cells, T cells, and also our immune memory. Certain pathogens, because of the way that they mutate, are very hard for our immune memory to be able to kind of keep up with. We looked at examples like the flu, right? The flu virus, the influenza virus, mutates very rapidly. And that's why, even if you got vaccinated against the flu this year, or you didn't get vaccinated and you actually caught the flu and you got sick and you got better, and you have that immune memory that you carry with you, it may be that by next year, right, the flu virus has mutated so that those certain antigens on its surface that your adaptive immune system did such a good job of selecting brand new naive T cells that happen to recognize them, right? And those eventually form the basis of not only saving your butt, right? And so that you didn't die from getting the flu, but more importantly, well, not more importantly, but just as importantly, right, those B cells and those IgGs stuck around as part of your memory. But maybe by next year, the flu virus has mutated so that those particular shapes, those antigens that you have good B cell memory of, right, are no longer are no longer what the virus presents. Right. And so that's why I said this before. Every year, the Centers for Disease Control, the CDC, they are aware, right, all over the world where certain outbreaks of different things like the flu are taking place throughout the year. And they can study what strains or what variations of the flu are the ones that are really beginning to pop up, and anticipate that those might be the strains that will end up hitting us, in several months. And so that's why they then formulate a revised version of the flu shot, right? And that every year it's important for you to get that new version of the flu shot to give you protection against those antigens, or recognition of those antigens that are the relevant ones for this year's version of the flu. Right? I'm sure all of you are aware in the news right now, there's a lot of talk about different variants of the SARS-CoV-2 virus that are popping up. It's the same thing, right? If you have a large population of people getting sick and harboring the virus, remember the human body, remember from the very beginning of the semester, we said the human body is itself an environment, right? An environment in which certain pathogens love to be. Right? We are a walking, talking, moving around environment. Right? So viruses, when they get in us, right, that's their ideal expected environment. And inside the body, they can mutate. Right? They can change. We already looked at examples of sort of scenarios where you might be in class and you get sick because classmates around you are coughing or sneezing with the flu or something like that. Right? You go home. You feel miserable for a few days. You catch up by binge watching Netflix. You catch up on all your favorite series. You drink lots of soup, right? But you get better. But if you return to class, right, you might end up getting your classmates sick, right? And then he or she, when she goes through that same convalescent phase of getting better, right, maybe in their body, the flu virus has changed just enough that when you both are back in class again and you're happy, right, she ends up giving you, right? A new version of the flu, and you get sick again. And it's this weird sort of back and forth, right? Um, so in those types of situations, the more reservoirs, host reservoirs there are involved, meaning the more people who catch the flu virus, right, the more chance that there'd be different mutations that end up getting spread. So that's what's happening right now with the COVID, right? The COVID-19. So the SARS-CoV-2, because there are so many millions of people who have it, each person's body represents the potential reservoir in which certain random mutations might happen to the virus. And that might result, right, in changes in the virus that make it more aggressive, more infectious, or other properties. Right? One thing students always ask me is they don't understand why it is that mutations always make things more aggressive. Right? If mutations are random, wouldn't there be a likely chance that in reality random mutations make the virus worse? Yeah, right? But the whole point is we don't care about those. Because if you have random mutations that make the virus less infectious, right, or less aggressive, what happens to that version of the virus? It just naturally dies out in the population, right? So the ones that emerge and come to the forefront are the ones, the random mutations that make the virus more aggressive, more highly infectious. And so now you hear about things like the UK variant, et cetera, and in the news, right? Different pockets are of outbreaks are happening where in a particular city or region, they're alarmed to find out that maybe like 70, 80% of all new COVID cases are a particular new strain or variation, right? So that's what we're talking about. That makes it very hard, right, for the immune system to be able to keep up with. Right? Some students have asked me, right, if you do get your COVID shots, right, the Pfizer, the Moderna, Johnson & Johnson, et cetera, are you still protected against these new variations, these new variants? Right? The short answer is yes, you still are protected because remember, right, in order for things like these viruses to still work and infect, right, they still have to have a functional glycoprotein spike on their surface that they use to bind to things like the receptors on the surface of your respiratory tract cells, and that's how they gain entry, right? So random mutations might alter the shape of that glycoprotein spike a little bit, right, to maybe make it, for example, fit better and bind tighter to host cells so that you end up getting more viruses that get into your cells and therefore it'd be more infectious, more aggressive, right? But it can't change that shape too much. Otherwise, it just won't work anymore. And then the virus won't be able to infect cells. So if you have been vaccinated using that glycoprotein spike as the learning tool for your immune system to select for different antibodies that bind that spike, right? Even if that spike mutates a little bit, right? Because you're talking about a handful of different antibodies that all bind to different parts of that spike, the different epitopes, right, that are part of the spike, right? Even if it changes and one or two of the antibodies that your immune system developed in response to the vaccine maybe don't work as well anymore, you would still have other antibodies as part of your memory that would still work, right? What that may mean is even though you are not as protected against the new variants because yeah, right, maybe the new variants can still infect you and make you feel sick. The point is because you have many antibodies that still do work against this variant, right? The extent of your illness, the severity of your illness, if you do catch the COVID, right, would be far less, right, less serious than if you never got the vaccination in the first place. So when you're reading about things, you know, it seems very alarmist in the news about now you're reading about people who were fully vaccinated and they're still catching COVID. What's that about, right? Keep in mind, right? It's not necessarily that the vaccine is going to 100% protect you from all the strains that are developing now, but they will provide enough protection. Your immune memory will provide enough protection that it'll make your experience of getting sick from the COVID far more tolerable, right? And it won't be as bad of a illness compared to if you did not get the vaccination in the first place. Right? Okay, let's look at another example here. So this is a classic example of pneumonia. So pneumonia can be caused by different causes. In general, pneumonia just refers to an infection of the lungs, right? Coughing and, heart, shortness of breath, things like that. Certain types of pneumonia are caused by bacteria. In particular, the Streptococcus genus of bacteria can give rise to pneumonia. Certain Streptococcus also give rise to strep throat, right? That's why they call it strep throat. Remember this, in order for your adaptive immune system to have good, solid, long-term immune memory against a particular pathogen, right, that's all based upon the presence of high quality, high affinity IgGs that your adaptive system learned to make the last time you faced that pathogen. And those high quality IgGs are in your blood, right? So that if the pathogen comes back again, right away, right, those high quality IgGs can bind to the pathogen and start the process of helping things like your innate immune system, the complement proteins and the macrophages, take care of the pathogen before the pathogen has a chance to really multiply a lot and make you super sick again, right? So if that's the basis of memory, then if you have pathogens that rarely mutate and rarely change the antigens that are on their surface, then you actually have the hope, right, of having not only long-term immunity against them, but as a species, we have the hope of eventually even eradicating or eliminating certain diseases. So things like smallpox, things like polio, right? Those are examples of pathogens, viruses that actually don't mutate very rapidly, right? They don't mutate nearly as easily or rapidly as something like the influenza or the flu virus, right? Or something like the SARS-CoV-2, apparently, right? As a result, after many decades of aggressive vaccination programs, where as kids, you get vaccinated against certain types of viruses like these, right? Because those viruses don't change that much in the population, after a while, we've pretty much gotten rid of things like polio and smallpox, right? However, there are also many examples of pathogens that do mutate a lot, right? And as a result, in the population, there are many different strains of them floating around, like the flu, like what seems to be sort of happening now with the COVID, and like this example here. So this is an example of Streptococcus pneumoniae, right? One of the primary bacterial causes of pneumonia. Right. So the Streptococcus pneumoniae has many different strains out there. There's something like 90 different strains out there right now. Each one over time had mutated to the point where certain antigens on its surface are quite different in shape. If you look at this example, so this is the bacteria over here. You can kind of tell on its surface, there's all these different proteins and things, right? Many of these provide the specific three-dimensional shape that your adaptive immune system would try to select antibodies against. But because there are so many variations of them, over 90 different variations, this cartoon on the bottom, obviously this is very simplified, right? But what it's trying to say is the same protein that's on the surface of all these different versions, strains of the pneumonia, are slightly different in shape, right? On this one, it's represented as a yellow triangle or an orange square, blue circle, etc., right? So that means that if you were infected with one of these strains at some point in your life, like the yellow triangle one, and you successfully selected for high quality IgGs as part of you getting sick and then recovering because you beat the infection, right? The IgGs that you have that recognize the yellow triangle, if a different strain then came along and infected you, like the orange square one, right, those IgGs might not be effective. Right? So we call these different strains of streptococcal pneumoniae, we call them different serotypes. What that means is that in the clinic, in the pathology clinic, if a patient presents in the walk-in clinic with pneumonia, right, and they take sort of a culture, a swab, and they try to determine, what this is, right? These different bacteria then can be identified, or these different strains of the same bacteria can be identified using different antibody tests, right? If you have different antibodies in the test that are specific to these different strains, then you can identify which one is infecting the patient. But you can see that makes it very hard for us to evolve as a species to have the ability to fully eradicate things like bacterial pneumonia, right? So that is why with things like pneumonia, bacterial pneumonia, it is quite possible to be repeatedly ill with bacterial pneumonia throughout your life, right? So this is one of those examples where I think a lot of people say, I don't get it, right? I thought the whole point was you have a good adaptive immune system so that if you get sick from something and you develop antibodies, then you're protected from it, right? So why is it that you can repeatedly catch pneumonia through your life? And have it be bacterial in origin. Right? It's because throughout your life, you might be exposed to one or more of these 90 different serotypes of bacterial pneumonia. And as a result, what this cartoon is showing here is even though you were infected with the yellow triangle version first, and you got better because you were able to select for B cells that make the yellow triangle detecting antibodies, so this yellow antibody here, right? And then in the future, you're protected because things like, these might be IgGs, are then used by other innate effector cells in the future so that if the yellow triangle bacterial strain or serotype comes up again, right away, you're protected. However, I think you guys all understand what I'm getting at here. If later on, a year from now, you are exposed to the blue circle serotype version of the bacteria, right, your yellow antibodies are no good here, right? And that means you're going to get sick again, and you have to start from square one and develop the blue antibodies, right? So consider this then. Why are diseases like pneumonia, why do they have the highest mortality then in infants and in the elderly, right? Well, hopefully we've learned enough this semester that you can already piece this together. You know that infants, when babies are first born, right, their immune system is still starting up. Right? Their immune systems rely heavily upon the IgGs that mom has transferred to them, right, before birth, and also after birth, the transfer of IgAs through breast milk. Right? But it takes several months for baby's own immune system to be able to start ramping up so that they can be producing their own selection of B cells and T cells. So therefore, if during that very vulnerable time, the first year of life, right, baby is exposed to a strain of, let's say, bacterial pneumonia that mom didn't have the previous memory of, then that can be very serious, right? And likewise, the opposite end of of life, right? In the elderly, we already learned that in the elderly, their thymus has really shrunken away, and by the time you're like in your 70s, right, most of your thymus is now just kind of fatty tissue. Some of the thymus is still there, right? So the elderly are still able to make some new T cells, because remember, that's where T cells are made, is in the thymus, right? So T cells are putting together their T cell receptors there, right? But the number of T cells, brand new T cells that are being made in the thymus of a 70-year-old pale in comparison, right, to the number of brand new T cells that are coming out of a teenager's thymus. Right? So that means in the elderly, you have reduced capacity to be able to come up with something new if your body for many decades didn't already have the memory of this, right? So that's why in the elderly, things like pneumonia can be so severe, because if they catch one of these different 90 serotypes, you know, I mean, that's the sad thing, right? If you live 90, let's say you live 80 years old, right? And during your life, you've been exposed to maybe 4 or 5 of these different streptococcal pneumoniae serotypes, and you came up with really good defenses against them, right? And those are the 4 or 5 that you have strong protection with. But by the time you're, you know, 80, right, you're exposed to a 6th type of this. And unfortunately, by then, the number of T cells that you're producing from your thymus, right, you have a very limited supply. Remember that it's really important to have T cells participating in your adaptive immune response, because T cells fundamentally are what help coordinate the actions of your B cells, right? So that is also why a couple of you have asked me, you know, if it turns out that in the elderly, they have a compromised or really worn down, worn out adaptive immune system because they no longer are producing a lot of T cells, why is it still useful or effective to immunize our senior citizens, our kupuna, right, with things like the COVID vaccine, right? Because after all, right, the first couple months of the whole tremendous effort to get people vaccinated was focused on our seniors, right? People who were 75 and above. So a couple of you asked me, I don't understand, because if they're no longer producing T cells, then what good is vaccinating them? It's not that they are no longer producing T cells, right? It's that their thymus has gotten to the point where they just produce very few T cells compared to when they were younger, right? So the vaccine is so important. Why? Because the vaccine allows their immune systems all the time it needs to be able to select from that limited pool of brand new T cells, which ones can actually recognize the SARS-CoV-2, right? And because it's a vaccine, right, they're not going to get sick with the SARS-CoV-2 as part of this learning the immune process, right? So hopefully you understand. That's why it's so critical that our senior citizens get the vaccine, because that buys them the valuable time, right, to still be able to come up with their own immune defense and memory, right? Okay. Here's another example of devious pathogens. So parasites have all kinds of tricks up their sleeve when it comes to evading the immune system. This example is particularly interesting. I think it's intriguing. So trypanosomes are a type of protozoan parasite. In other words, they are single-celled organisms. Here's an image of it. So here are your red blood cells, right? And these little sort of roughly looking things are these trypanosomes, right? They are single cells, but they are infecting the blood here. And trypanosomes, such as Trypanosome brucei, this is the cause of what we call sleeping sickness. So in places in Africa, sub-Saharan Africa, sleeping sickness is a very big public health problem. They call it sleeping sickness because the person who is infected with it and gets ill with it gets increasingly more and more fatigued, right? To the point where they're just so debilitated and tired that they essentially sleep nonstop, right? And if it goes untreated, right, they basically end up just dying due to fatigue, right? So it's caused by this parasite, this trypanosome. The reason why this trypanosome is so devious is that, you know, on its surface, of course, it has to make many different types of proteins as part of its normal structure, right? And so many of these proteins on its surface, you would say, yeah, I bet you some of these serve as good antigenic signals for the human immune system, the adaptive immune system, to learn from, right? But this is where things get really interesting, right? Certain of its membrane proteins, right, are coded for not just by one gene, but by 1,000 different genes that code for, right, a particular surface protein, right, or group of surface proteins. And at any given time, the trypanosome will be using one of these 1,000 or so genes as the one to make the protein on its surface. So we call these VSGs, or variable surface glycoproteins. And the parasite has the ability to switch between these different genes in order to evade the immune system. So look here. So let's imagine that this is the gene for this particular VSG protein that the trypanosome needs to make in order to have this protein on its surface. And so at this time, this red version of the gene is the one that's actually being expressed. Okay. But you see here, all these different colored blocks, A, B, C, D, E, F, whatever, right? These are different variations of the same gene, each one a different copy. And it has like 1,000 of them. So what it can do is at any time, it can swap out which one of these genes gets expressed. So here over time, it started making the red version of the protein, but then the trypanosome swaps it out, right? It takes the yellow version of the gene and actually through a process of gene conversion that is physically replacing what copy of the gene is in the expressed region of its DNA, right? And now expresses the yellow version, right? And then later it can convert again, and now maybe it puts the blue version, right? Because the trypanosome always needs to work at having all these proteins, right? It needs to have these proteins as part of its structure. But the point is, it can go ahead and change the version of the proteins many times. So as a result, on the right, what we see is the clinical course of trypanosome infection. On the x-axis is time in weeks from the time of infection out to, let's say, 4 weeks out, right? And on the y-axis is the titer, or the amount of parasite in the patient's body. Right? So let's say this trypanosome worm starts off by expressing the red version of this membrane protein. So as it infects, right, the numbers of the parasite increase in the patient's body until after a week or so, when the patient's adaptive immune system begins to kick in, right? And it selects for naive B cells that have IGs that happen to have the right fit, that recognize the red version of this trypanosome protein. So the dotted line represents the sudden increase in levels of anti-VSG, A antibody, right? But once that's happening, look what happens, right? It kills off the trypanosome, right? So the levels of the parasite begin to fall off as the adaptive system is being successful. But then what happens? At a certain point in the background, right, a number of these trypanosomes in the body have already switched. They're now switching to the yellow version, right? So that version, those trypanosomes begin to multiply, right? And the levels of parasite go up. Then the patient's adaptive system has to play catch up, right? Now it has to select for antibodies against this version of the protein, right? And as it's doing that, and it's successful in getting rid of these parasites, in the background, the same thing is happening over and over and over again, right? So instead of having your normal adaptive immune response, where you have the big peak, this is your primary immune response, right? Your primary exposure. So you have your big peak in B cell activity and T cell activity that then begins to taper off, right? What the parasite is doing is keeping the adaptive immune system constantly maxed out, trying to keep up with it. And the toll that it takes on the body then, right, is it really wears down the resources of the body, resulting in increased fatigue, and eventually, right, unless it's treated, the immune system is just going to lose, right, and the patient's going to die. Here's another example. So that was a parasite, right? What about some more examples of viruses? So the herpes virus. Herpes virus is a big family of viruses. You might not be aware, but things like cold sores, right? So the herpes simplex virus is the cause of cold sores. We've all had cold sores before, right? That's when in the corner of your mouth, you get that kind of painful little red lesion, or maybe on your lip up here, right? And, you're kind of like, it hurts, it's sore, but really it's more of like an embarrassment, right? Because you don't walk around, you have a big cold sore on your lip, right? So that's caused by a herpes simplex virus, right? And just with like any other viruses, if you think about it, in order for a viral infection to be completely cleared from your body, right, your adaptive system, right, must have really good CD8 cytotoxic T cells that have been activated, and they're going around and they're bumping into any of your body cells that are showing evidence of being infected by the virus. And the cytotoxic T cells tell those infected cells, right, undergo apoptosis, eliminate yourself, before you spread any more of these virus particles, right? So that is a critical part of really eliminating virus from the body, is having good CD8 cytotoxic T cells that must be able to see the little peptides that are coming from the virus being presented on MHC proteins on the surface of the infected cell, right? So herpes simplex virus, the cold sore virus, it infects the epithelium. So for example, that's why you have the little sores around your lip, for example. But the virus can actually spread to your nerves. It can get into your neurons, your nerve cells, and it can actually travel up those nerve cells to places like the ganglia of your brain. Now, that might sound really scary, right? Because now you have a viral infection in your brain. But this is where things get interesting, because these neurons, neurons are one of a few examples of cells in our body that don't actually express high levels of MHC platforms on their surface. I actually don't know why that is. I think it probably has to do with the fact that the function of neurons in terms of transmitting, electrical impulses may be somehow compromised if they have a lot of MHC proteins on their surface. I don't know for sure, right? But the bottom line is, so these neurons, right, are a good place for these virus particles to kind of hide out. Because if the neurons aren't really actively expressing MHC platforms on their surface, then they're not actually going to be displaying peptides from the virus that's coming from inside of them. And therefore, they will be missed by things like patrolling CD8 T cells, right? I mean, actually, come to think of it, now I remember, I think one of the main reasons evolutionarily that our nerve cells don't express a lot of MHC, and especially MHC1, is because, we cannot really afford to have our nerve cells be targeted for destruction by our CD8 cytotoxic T cells, right? Because many of our nerve cells, you have what you have, and they don't actually regrow, right? So you can imagine if you had a bad situation where you have cytotoxic T cells killing your nerve cells, that couldn't be good, right? So you have these herpes simplex viruses hiding out in the nerve cells because the nerve cells are not alerting your immune system to the fact that they're harboring these viruses. And that sounds really scary because now they've gotten up into your brain. But the thing is, the catch here is those nerve cells are also not the optimal cell in which the virus particles themselves can multiply, right? So in other words, the neurons don't participate in really spreading the virus. It's just a place where they're hiding out, and the virus that's inside the nerve cells don't multiply. They just kind of hang out there, right? Until something happens. So that something that happens might be certain changes physiologically in your body's chemistry that are brought upon by like stress or some other type of illness or different types of hormonal shifts, right? Those types of things might actually encourage the virus particles to kind of reactivate, at which point they may migrate down the neurons and re-enter things like epithelial cells. And that's why you might get cold sores coming out again when you're stressed out or depending upon different types of hormonal cycles, right? So chickenpox is another good example of a herpes virus that does this. So when you catch chickenpox as a kid, I think we discussed this before, right? It used to be when I was a kid that we didn't have a vaccine against chickenpox. The vaccine for chickenpox, I think, was introduced in the early to mid 1990s. So I think most of you have had chickenpox vaccination, right? But back when I was a kid, you know, parents handled it the way Mother Nature intended it to be handled, right? Which is, in order to get immune memory, you have to be exposed to it. And so I told you about how during the summer, especially when there's no school going on, if one of the neighborhood kids got chickenpox, then the parents would get together and have this sort of like chickenpox party, right? Where they throw all the kids together and we play together, and then we all come home with chickenpox. And then we have all these bumpy sort of itchy pox, you know, lesions for like a couple weeks. But the whole point was it's during the summer, so we're not missing school, right? That's why the parents wanted to do it that way, right? But chickenpox, after you've had chickenpox, you've developed adaptive immune memory against it, right? So it's very rare to actually catch chickenpox a second time. Chickenpox is one of those examples of you catch it once when you're a kid, right? And you're pretty much protected for life. Right? Or you're vaccinated as a kid and you're pretty much protected for life. However, when chickenpox just causes itchy like blisters and lesions in kids, when chickenpox infects adults, it can be a lot worse. Because again, just like this other example of cold sore viruses, the chickenpox virus can actually hang out in your nerves, right? And hang out there indefinitely. So even though I caught chickenpox as a kid, and I've had immunity my whole life, it actually does not mean that my body is free of chickenpox viruses, right? And you say, wow, that's crazy. Yeah, I probably have chickenpox virus inside, you know, some of my nerve cells and things like that. The whole point is, though, is when that chickenpox virus starts getting rowdy and wakes up again, I have very strong adaptive immune memory, right? So right away, as the virus begins to reappear, right, my adaptive immune system shuts it down again. Right? I have cytotoxic T cells, I have IG against it, and it almost like chases the chickenpox back into the neurons again, right? Into hiding, right? But the problem is when you actually have chickenpox exposure for the first time as an adult, right? The chickenpox really gets into your nerves and it can cause some very painful neurological side effects. And so in adults, we don't really call it chickenpox. We actually call it shingles, right? And it can be very painful. But that's another reason why it's important for you to get the chickenpox vaccine as a kid, since because it's available, right? All right. So this table is a summary of different examples of how viruses have evolved, many of these very clever, insidious ways of interfering or eluding our immune system. Right? So it's another example of one of those things where I don't expect you to memorize it, but I would like it if you spent a few minutes and just read it, okay? Because it's not hard to understand. I mean, it might be intimidating because there's a lot of boxes with words, but whatever, right? I don't expect you to memorize this, but if you read this, I guarantee you'll really get you thinking about all these different ways that viruses can interfere with our immune system, right? Everything from being able to inhibit or mess with our ability of complement proteins to work, or inhibiting our inflammatory responses. Because remember, our inflammatory responses are a very important part of signaling for a very strong, robust adaptive immune system, immune response, right? Blocking of cells from being able to process and present antigens, and also suppressing different types of immunology signaling going on between cells, like messing with cytokine signals. Okay, so take a few minutes and take a look at this. I think you'll find it interesting. All right. So the last 2-3 slides, I want to give you some examples of other really, I think they're cool, but only because they're very, very sort of like evil and insidious ways that pathogens have come up with to kind of evade our immune system. So certain bacterial proteins are very effective in disabling the immune system. A group of these proteins function as what we call super antigens, right? So the name super antigen, right? You say that's kind of weird. It makes it sound like this is a really good antigen, right? Well, that's exactly what it means. But you might say, well, wait, a super antigen, it says here, intentionally stimulates T cell activation, right? You're like, that doesn't make sense, because it seems like the last thing that a bacteria would want to do is actually stimulate a lot of T cell activation, right? But this is where things get really kind of clever. The type of T cell activation that these superantigen proteins stimulate is non-specific T cell activation. So let's look at this figure, this cartoon. All right, so here we have a T cell. It's an alpha beta T cell, so it's part of our adaptive immune system. This T cell is kind of floating around somewhere. Here's an antigen presenting cell. All right, in this case, it has MHC class 2, right? So this would be a helper T cell down here, right? A CD4 positive helper T cell. And this red dot is just any old antigen that's being presented by this particular antigen presenting cell, right? So this red dot might have nothing to do with the bacteria at all. The bacteria that's floating around somewhere, not in this cartoon, is making this blue blob, which is the superantigen. What the superantigen does is it's the right shape to fit and bind to the MHC class 2 on one side, and it's the right shape to bind to alpha beta T cell receptor on the other side, therefore bringing the two together, right? Now you might say, well, yeah, but that's how it works, right? If it's actually bringing together the T cell receptor with the MHC, then isn't that how, the T cell is supposed to activate? But not so fast, because remember I said this red dot that's being presented on the MHC can be any old peptide, right? It's not the peptide that is actually the right shape for this T cell receptor to bind to. So if this superantigen is going around just activating any CD4 T cell, right, regardless of whether that CD4 T cell has the right T cell receptor to be of any use, right? And of course, the antigen presenting cell is presenting B7 because there is an infection going on, right? The bacteria that's making this superantigen is infecting the tissue. So the naive T cell here gets that combination of T cell receptor, CD4, and B7 CD28. So it activates, right? But because this is any old helper T cell, what happens is you have a tremendous number of CD4 helper T cells that activate improperly. Right? So essentially, the superantigens are overloading the immune system. There's non-specific responses. Up to 20% of all CD4 T cells that happen to bump into this superantigen will activate, right? So it causes a huge flood of confusing cytokine messages going on, right? And you have so many of these helper T cells just activated, right? There's just so much noise going on that the actual bacteria is managing to kind of stay below the radar, right? So it can escape from being specifically detected. So here it is, here's the structure. All right, the superantigen over here is the blue. Here's the MHC in green and yellow, just like the cartoon. Right here is the alpha beta T cell receptor down below these parts. Right, so in this case, this alpha beta T cell receptor should not have bound to this MHC presenting this peptide here, but the superantigen is making it. Right, and so it's just like plugging in too many things to an outlet. Right, you're going to overload the system. And because of that overload, the bacteria actually benefits. All right. Many food poisoning is due to superantigens. So certain bacteria that you end up eating as part of your food, like salmonella, things like that, it produces superantigens. And that's why oftentimes food poisoning can be such a severe, quick response, right? Not quick onset, but within maybe like 2 hours after eating bad food, right? You start feeling, oh, that's not good. And then later in the evening, boy, you're a big mess, right? Because your immune system is overloaded and is freaking out due to the superantigen activity. All right, this is the last slide. Another example are things like bacterial proteins that can interfere with the function of antibodies. So normally, this top part of this cartoon here, you have a particular bacteria, right? If you have an IgA, for example, in your mucosal layer that recognizes the little red bumps on the bacteria, right? Then the IgA might bind to it. And then you have something like a macrophage that has Fc alpha receptor that can hang on to the stems of the antibody, making it way easier for the macrophage to take in a lot of the bacteria and destroy it, right? So one type of superantigen sort of like protein we call SSLP7, or staphylococcal superantigen-like protein, actually binds to the IgA. So here you see in the lower figure, you have this SSLP7, right? It binds to the stem of the IgA, and therefore the stem of the IgA is no longer available to be picked up on by the macrophage. Right? At the same time, the other end of the SSLP7 binds to complement protein C5. Right? So you end up creating a shield around the bacteria, because remember, this is not drawn to scale, right? The surface of bacteria would basically turn blue because it'd be studded with these SSLP7s that are holding C5 as sort of like a shell around it, right? Why would it do that? Because if you have a shell of C5 being kept away from the surface of the bacteria, right, then more C5 cannot come in and actually participate in normal complement activity, right? So you're essentially using complement proteins to create a shield to prevent other complement proteins from coming in and attacking the bacteria, right? I think that's wonderfully devious, right? And another example of these clever ways that pathogens have come up with to evade our normal immune response. Okay, I'm going to go ahead and stop there.